So Zoom is on, right? Zoom is on, okay. Well, we'll get started. Actually today, it's my great pleasure to introduce both Chenjie, who will introduce Dr. Gail Mandel, but I just want to say one thing. I'm very grateful to Gail, not only coming, but mentoring me since I was in Buffalo. She actually was the outside committee member of my thesis. Without her signature, I wouldn't be here. I actually wouldn't get my Ph.D.
50 years ago.
So thank you for believing in me, and I finally have the opportunity to welcome you to MIT to show us the great work you have been done since I was kindergarten. No, you're not that old. Sorry. So Chenjie and I jointly invite, and thanks to Simon Center, Baganka's support, and Chenjie is a postdoc in my lab. He has been, since joining my lab, has been working on Rett syndrome, and he did his PhD in Zhejiang University, which I also graduated from Zhejiang University. So Chenjie will give an introduction to welcome Gail.
Thank you, Guoping. It's my great honor to introduce today's speaker, Dr. Gail Mandel. Dr. Mandel got her PhD in immunology from UCLA and pursued postdoc training at both UCLA and UCSD. In 1984, she established her own lab at Tufts Medical Schools as assistant professor with primary focus on the I/O channels. So back to 1980s, Dr. Gail Mandel is one of the pioneering who successfully cloning the voltage-gated sodium channels from Drosophila. And also, she generally shows all this cDNA to numbers of labs, which makes scientific progress in the sodium channels fields. So after that, Dr. Gail Mandel started to become interested in how regulate elements, for example, promoter and enhancer, regulate the sodium channel expression in different tissues. So one of their works actually identified negative repressors of the major brain sodium channels. And this works further lead to a breakthrough discovery in 1995 when two independent groups, one is from Dr. Gail Mandel's lab and the other one is from David Anderson labs, they identified repressor binding proteins called RAS, which is a transcription repressor that silences the neuronal gene expressions in non-neuronal cells. And her subsequent works on RAS have revealed that RAS is also playing important roles in the neuronal differentiation, and also dysfunction of RAS also has been observed in human agents. So because of her background and also expertise in the transcription repressions, so during the past few years, Dr. Gail Mandel started shifting their focus on other transcription repressors. So one of them is called MECP2, a gene mutations on these genes called the Rett syndromes in humans. So her works on Rett syndrome actually revealed that glial cells, like neurons, are a key component of the pathogenesis of the disease. So today, Dr. Gail Mandel will present their recent works on developing potential therapeutics targeting Rett syndrome. So please join me to welcome Dr. Gail Mandel for her talk today.
Yeah, thanks very much for that nice introduction, Chenjie, and for the invitation to speak here today. As Chenjie mentioned, I was an assistant professor actually I was an instructor first at Harvard and then an assistant professor at Tufts, and I've maintained a lot of friendships with people that I worked with then to today, so I really enjoy coming back to Boston. So what you're looking at is a picture. The OHSU campus is split between the waterfront and up on a hill, and there's a tram that can take you between both places. The perspective is a little off because that's Mount Hood on the bottom, and the tram is not higher than Mount Hood, but there is a nice view from the tram. And I want to disclose that I'm a co-founder and shareholder of Ecotherapeutics, a neurology-focused genetic medicine company. However, my lab is funded completely by NIH, and all of my data is, of course, open to the public. So today I want to talk about 4 topics. First, for people who aren't familiar with Rett syndrome, I want to give a brief introduction to the disease and also talk about MECP2, which is mutations in which give rise to this disorder. And then I'll tell you about our efforts to use targeted RNA editing to recode MECP2 mutations, and then discuss a proof of principle for RNA editing in non-dividing neurons in culture, and then our work to apply RNA editing in mice and look for alleviation of key Rett neurological phenotypes. And then in the end, if there's time, we can have a discussion about challenges and open questions. So the root cause of Rett syndrome, as I said, is mutations in MECP2, and that was determined in a positional cloning experiment by Huda Zogby in 1999. Because MECP2 is on the X chromosome, and because of dosage compensation, females are mosaic for loss of MECP2 in this disease. The lifespan of females with Rett is about 60 years. And males, these are de novo mutations which give rise to this disease. It's bad luck, so both females and males can get the disease. And in males, the disease, because they only have a single X chromosome, the disease is more serious, and males usually die before the age of 2 by an encephalopathy. Rett has been described in stages clinically. So the first stage is an overtly normal development for 16, 18 months, and then there's a crisis. And that crisis is followed by regression of milestones, which are typically, obviously, loss of speech and loss of purposeful hand motions. Then there's a progression of motor disturbances characterized by repetitive hand motions, shaking of torso and limbs, irregular breathing, which is quite prominent with breath holds, seizures, and immobility. And after that, the symptoms stabilize. And like other neurological diseases, mouse models have been very useful in helping to understand some aspects of the disease and its link to MECP2. So in the female mouse models, they live a normal lifespan. They have latent symptoms like the humans do. They're overtly normal for 4 to 6 months, at which time they start to have progressive motor disturbances, tremors, irregular breathing, and apneas. And I want to say that the breathing irregularities and the apneas are very similar to what's seen in the human females. And then the symptoms stabilize. In contrast, the mouse model for males is quite different. They undergo premature lethality after about 3 to 4 months. They do have latent symptoms, but they're overtly normal for 4 to 6 weeks compared to 4 to 6 months in the females. Then there's progressive motor disturbances, tremors, irregular breathing, apneas, and immobility, and the symptoms progress in this case because they die prematurely. And the reason for premature lethality in the males is not known. So we know quite a bit about the biochemistry of MECP2. MECP2 was, let me see if I get my pointer going here. MECP2 was originally purified in the lab of Adrian Bird on its basis of binding to methylated CG dinucleotides in the genome. He then determined that MECP2 has a modular structure, a portion of the molecule that binds to methylated DNA, and then a portion of the molecule which recruits a co-repressor complex that involves histone deacetylase. So the model that he proposed, which I think still stands today, is that MECP2 is a bridge between 2 repressor mechanisms. methylation of DNA on the one hand, and histone deacetylase repression on the other. Now, because MECP2 is associated with developmental delay and intellectual disability, the MECP2 gene is frequently screened for mutations in clinical cases of developmental delay. And if you put the mutations together, you can see a pattern for where these mutations reside in the protein. And you can see by the red vertical lines, which are the missense and nonsense mutations, that they're not randomly occurring through the protein. They cluster in these two functional, primarily in these two functional domains. In contrast, benign polymorphisms in the population are broadly distributed throughout the protein, and they mostly occur not where the missense and nonsense mutations occur. And the missense and nonsense mutations represent about 60% of the mutations that give rise to Rett. Now, despite these pervasive mutations, if you express MECP2 in a null background postnatally, you can reverse many of these symptoms. And this was shown in a seminal experiment by Adrian Bird's lab in 2007. And the strategy was a LOXP conditional Cree deleter strategy, and it involves the insertion of a stop sequence upstream of the functional part of the protein. So in the presence of the stop sequence, MECP2 is non-functional. And then when the stop sequence is excised by the Cree deleter in response to treatment with tamoxifen, the stop sequence is removed and you end up with a normal MECP2 protein. So in the middle panel here, I'm showing the time course of the acquisition of symptoms in male mice. And actually, I should say that for the entire talk, I'm only going to be showing data from male mice. And the reason is that for many proof of principle experiments, it's really nice to use the males because they develop symptoms so quickly, within 4 weeks or so you don't have to wait 4 to 8 months before you can begin assessing the behaviors. But in most cases, although we start with males, we also ultimately look at the females. And that's true for this experiment that Adrian's lab did. So this is a postnatal experiment, and in males, symptom onset is between 4 and 7 weeks, the phenotypes that I talked about earlier. And then this particular case I'm showing, tamoxifen is added at a point where advanced symptoms in the mice are starting to be apparent. And then eventually, as I said, there's premature lethality. So if you look below at the left graph, what's being plotted is an aggregate phenotype score. And the phenotypes that are being scored are mobility, gait, hind limb clasping, tremor, irregular breathing, and overall general condition. This is kind of a subjective scoring, observational scoring system that's used by many labs. And the way the scoring works is that each of these phenotypes get a 0, 1, or 2 for a score. And 1 is usually wild type, and 12 is usually the animals are dead by 12. And in fact, often we euthanize the animals before that for humane reasons. In my lab, we do it if the mice have lost 10% of their body weight. So in the case where the stop sequence is present, you see there's a rapid transition to a high phenotype. And in the case where the mice have been treated with tamoxifen, they originally start to develop symptoms like the stop mice, but then the tamoxifen kicks in, they get a little bit better, and they plateau at a level that's not quite wild type, but pretty good. And then this graph here is a survival curve showing that the premature survival travels with the untreated stop Cree mice, but the mice that are treated with tamoxifen, at least in this experiment, 4 out of 6 still living, and they live a normal lifetime. And this experiment was also done in female mice with similar findings. So this experiment, I would say, kind of galvanized the field into thinking about the possibility of reversing at least some or alleviating symptoms in Rett syndrome. And one thing that, of course, we can't do this experiment in people, but one thing that was really important in this experiment in mice is that the removal of the stop sequence allowed the protein to be expressed under its normal regulatory control mechanisms, promoter and enhancers. And that's important because there's also another disease, an MECP2 duplication disease, which has its own set of severe neurological symptoms. So the MECP2 seems to be a gene that's very finely titrated in terms of level. So the therapeutic window, in some sense, is only twofold. So you can imagine several approaches which would allow you to maintain MECP2 at physiological levels without having overexpression. One is to activate MECP2 in the inactive X chromosome. And in fact, Rudolf Jaenisch and collaborators recently published a very nice paper looking at human neurons in culture and showed that by interfering with epigenetics, they were able to see reactivation of MECP2. And furthermore, that reactivation seemed to correct the smaller size of neurons that occur in Rett, as well as correcting the process complexity and length. So another thing I want to say is probably a feature that contributes to the reversal in this disease is that the cells don't die. There's no obvious cell death in the brain cells. They become stunted. The girls are slightly microcephalic, as are the mouse models. But it's as if the chromatin is just sitting there waiting for MECP2. I sort of think about it like they're hibernating or something. And then you add MECP2 back, and surprisingly, they're receptive, and it seems to restore whatever their problem is in its absence. And one conundrum is that we still don't understand really why the loss of MECP2 results in this disease, because the transcriptional changes are broad, but very small, less than 1.5-fold. And so this is still a little bit mysterious. So in addition to thinking about activating MECP2 on the inactive X, two other approaches that are obvious, especially to this audience, I'm sure, is RNA and genomic base editing. And I was originally attracted to RNA editing because I liked the idea that it could be transient in case there are off targets. It could be tunable, and it could be, depending on the context, reversible. So for those of you who may not be acquainted with A to I to editing, and the type of editing that I was interested in particular was adenosine to inosine RNA editing. This is a post-transcriptional mechanism, and it's very prominent in cells. And what happens is there's a hydrolytic deamination of adenosine, which creates inosine. And because inosine base pairs with cytidine, it's treated often by the cells, not always, but often as guanosine. And the ability to make this substitution can have profound effects on the cell. It can affect splicing, it can affect microRNA processing, it can recode proteins and change their function, diversify their function, and other things as well. So I was also interested in adenosine to inosine editing when I looked at the types of mutations that were the most common in Rett. And that's shown here. So again, I'm showing you the modular structure. And these mutations represent some of the more common mutations. The ones that I've highlighted in yellow here are G to A mutations. And in addition, there can be C to T mutations, which create nonsense mutations. And these four, 168, 255, 270, and 294, represent about 30% of the mutations that give rise to Rett. And altogether, it's about 40% of the mutations. So the idea then is to try to target RNA editing to reverse adenosine or change adenosine back to guanosine. I also want to point out that for these nonsense mutations, if we were able to to change adenosine back to guanosine, we won't get back the same amino acid. It'll be a tryptophan instead of an arginine. So Jackson Labs has been generating mice that have all of the mutations as well as all the tryptophan substitutions. And so far as we can tell with confidence, all of them except R168 tolerate the tryptophan at that position. The data isn't out yet in 168. And I can say that Guoping's lab and my lab in our own hands have looked at 270X and 270 with the tryptophan, and we both, even though we made the mutations different ways, agree that the tryptophan is well tolerated in this molecule. So how do we turn this into a therapeutic? So one thing that's known is that there are 3 enzymes, adenosine deaminases acting on RNA, which have the ability to deaminate adenosines. It's a family of proteins, and their substrate is double-stranded RNA. And what I'm showing you here are the conserved functional domains in this family. So ADAR1 has 3 regions that recognize double-stranded RNA and an orange catalytic domain. And ADAR1 likes to edit repetitive elements. And in humans, that's mainly ALU sequences, there's hundreds of millions of them, and millions of them are edited by ADAR1. And the idea is that ADAR1 protects against a cell not recognizing the repetitive elements as non-self. And in fact, an ADAR1 knockout gives rise to an autoimmunity problem. ADAR2 also has a catalytic domain and has 2 regions that bind to double-stranded RNA. It has many, many fewer substrates. In fact, only a few dozen, and many of those happen to be proteins that are involved in neurotransmission. ADAR3 lacks a catalytic domain, but has regions that bind to double-stranded RNA, and it's been considered that it might act as a dominant negative to the other 2 ADAR forms. So actually, the first experiment to consider RNA editing, targeted RNA editing to an unnatural substrate as a therapeutic was in 1995 by Wolf and colleagues. And Wolf and his group had the idea to use a guide to recruit ADARs to an unnatural target and use the guide to make the target double-stranded. And he tested this idea in Xenopus oocytes, which at that time was a common heterologous system used. And he did see on-target editing of his substrate, but also it wasn't that specific. There were off-targets. And either because of that reason or for some other reason, this idea of making a therapeutic from ADARs was essentially ignored for 17 years. And then in 2012, Thorsten Staffers at the University of Tübingen resurrected the idea of using a guide to target ADARs to an unnatural substrate. And his idea was to actually covalently link ADAR to the guide. Then a year later, Josh Rosenthal's group at the University of Puerto Rico also resurrected the idea of a guide, but not a covalent guide. And I happened to visit Josh in 2014 in Puerto Rico, and he showed me his beautiful work targeting ADAR to mutations in a chloride channel which give rise to cystic fibrosis. And I thought that was quite interesting. And so I decided to look at using the same approach to correct mutations in MECP2. And we used his system that, we used Josh's system that he had developed, and it's shown here. And the idea was to get rid of the regions that naturally recognize double-stranded RNA of its normal targets, and instead fuse a peptide from bacteriophage lambda, which binds to its own hairpin called the lambda box B hairpin with very high affinity, at least in vitro. And so in our case, the MECP2 guide would contain a lambda box B hairpin and sequences surrounding the target A. And we have a mismatch between A and C because endogenous ADARs happen to like a mismatch at the target A. And we used a hyperactive form of the catalytic domain. This mutation was made in Brenda Bass's lab in 2012. And Josh used it, we used it, Feng eventually used it also, I think with the idea that if we couldn't get it to work with the hyperactive domain, it wasn't going to work with the natural domain. And so the first question we wanted to ask was whether we could repair MECP2 and non-dividing neurons. And that was because all previous experiments had been done either in Xenopus oocytes or in dividing cells. And our strategy was to make a mouse model with a patient mutation, shown here. This is a mutation that had been characterized before biochemically. It's in the DNA binding domain of MECP2, and it loses, it severely loses the ability to bind to DNA, and also it's very unstable. And so we thought this was good to start, even though this is a rare mutation, we thought we would start with it because it would make it easier to see recovery if the repair is successful. And our first experiment used recombinant AAV12 that included the hyperactive catalytic domain with the lambda peptide, so it's a fusion protein. We put in a nuclear localization signal because ADAR2 primarily works in the nucleus. We used a neuronal promoter for synapsin 1. And on the same AAV, we had an RNA guide for MECP2 under control of the U6 promoter, or a scrambled guide instead of MECP2. We transduced the neurons with these one or the other viruses. We waited 7 days. Then we did Sanger sequencing for editing efficiency. We measured MECP2 protein expression, and then we developed a single-cell assay to look at DNA binding ability. We actually were able to see very high efficiency of A to I editing, as shown here. And importantly, the editing was dependent on the presence of the MECP2 guide. We didn't see any editing with a scrambled control. And then we looked at MECP2 protein levels, and we saw that there was about 3.5-fold increase in protein when we transduced with the MECP2 guide. And I think this is probably the reason it isn't higher, given the editing, I think is because we didn't wait that long in culture for MECP2 to be made. Okay, so then we wanted to see, okay, we've made protein. Now, does it bind to DNA? And we wanted to do this in an in vivo assay. And so we used confocal microscopy in our purified hippocampal neurons. And we labeled for the editase with an epitope, so it'd be an antibody to the epitope because we weren't sure about the quality of the antibodies for that ADAR2 catalytic domain yet. We used DAPI that labels nuclei, and you see how there are these dots here? That's because mouse cells have foci of condensed heterochromatin in the cells, which happen to be very enriched in methyl CG dinucleotides, which is where MECP2 likes to bind. So this top panel shows the pattern for non-injected wild-type MECP2 cells. These are the dots of heterochromatin. You can see by DAPI they stain very darkly, and you can see that MECP2 likes to co-localize with these dots. And then the next two panels represent our mouse model, neurons from the mouse model that are transduced with editase in the scrambled guide or editase with the MECP2 guide. And the confocal microscopy was done by a really good histologist, Jenna Fisk. And in the middle panel, you can see with the scrambled guide that there is a little bit of MECP2 present in the nucleus. It's not a complete null, but there's no association of MECP2 with the puncta. And we've never seen, actually, in this assay, association of a mutant or a scrambled guide cell associating with the heterochromatin. And in contrast, if you look at the bottom panel, you can see that there's more protein more MECP2 protein in the nuclei of the cells. They got the MECP2 guide in the editase, and it seems to associate quite well with the puncta. And the quantitation here shows that the number of DAPI positive and editase positive cells in this assay were the same between the scrambled and the MECP2 guide. But we only saw, as I said, MECP2 association with heterochromatin in the cells that got the guide. It was guide dependent. In this experiment, we didn't do a whole transcriptome RNA-seq analysis. We only looked for bystander off-targets within the guide region. We found one off-target site which we could eliminate with a mismatch. So while we succeeded in showing that non-dividing cells were capable of targeted RNA editing, we wanted to do a more challenging experiment, which was to ask about the in vivo situation. We wanted to look at at least three different populations of neurons in the brain and ask whether all of them were amenable to RNA editing, or might there be RNA binding proteins, for example, that would change the accessibility of RNA of the target to RNA editing. And so we switched our paradigm a little bit, and we used the same. We made two new viruses, but we used the same editing components that I showed you before. But this time we took advantage of some creative work by Benjamin Deverman when he was in Gradnaru's lab in finding a new serotype of AAV9, PHP.B. Did I write that right? Or is that a capital B? I can never remember. And what we decided to do was to use stereotaxic injection of the viruses directly into the hippocampus in P28 to P35 mice, wait 4 weeks for expression, then do immunolabeling for the editase. This is really a control experiment to make sure that our injections work. Then we were going to do Sanger and whole transcriptome RNA sequence analysis to look at on-target and off-target editing, and then quantitate MECP2 binding to heterochromatin. And I should mention, this is, we're not really measuring MECP2 binding. It's a proxy for MECP2 binding because we're only measuring association with the heterochromatin. But many experiments have shown that it's actually a really good proxy when you compare it to biochemical experiments. Okay, so this is the control experiment. This is a section stained with antibody to HA, which measures editase, and you can see there's nice editase expression throughout the hippocampus in the different regions. This is just DAPI. And when we looked at percent editase positive and DAPI positive cells, there was no difference in any of these three populations between the non-targeting guide and the targeting guide. Then our strategy was to laser capture each of these neuronal populations, make cDNA, and do Sanger sequencing to look at on-target editing. And the results are shown here. And we were happy to see very robust RNA editing in all three populations. And again, the editing is dependent on the presence of the targeting guide. We then did transcription-wide RNA analysis and unfortunately identified problematic off-target events. In fact, there were 3,000 of them compared to non-injected controls. And then below, I'm plotting the percentage of total sites versus the average editing percentage per site. This is from 3 mice, and we required that all 3 mice had the same editing events to be counted, and we also subtracted any polymorphisms or any editing sites that were present in the controls, which were presumably due to endogenous ADAR. So what you can see is that the editing is graded. So some events are edited more highly than others, and about 50% of the editing events are only edited between 10 and 20%, which is to say that 80% of an RNA at a particular site it did not have the off-target. Nonetheless, this is a lot of off-targets and obviously has to be taken account. And we asked whether these off-target events would prevent association of MECP2 to the heterochromin. So this is another confocal experiment like I showed you before. The wild-type non-injected cells, non-targeting guide, targeting guide. And in this case, the non-targeting guide with editase looks just like a null MECP2 condition. We really couldn't see any MECP2, but in the mice that were transduced with the editase, you can see there's recovery of the MECP2 protein, and again, it's associated with the heterochromatin. The quantitation is showed on the left here. This is a representative count from the CA3 neurons, wild-type condition, non-targeting condition, targeting condition. And you can notice a couple things. One thing is there's a really big distribution of the MECP2 intensity in heterochromatin, even in the control wild-type cells. It's about twofold. And this is something we predicted based on some earlier experiments that said different cell types in the brain express different amounts of MECP2. You can clearly see this in a single cell analysis. This is from 2 mice, 20 cells per mice, so each of these distributions has 40 cells. Then the non-targeting guide still has a little bit of a distribution, but clearly distinctly different from the wild type. And the targeting guide cells were sort of intermediate. And what we were pleased to see is that the estimates of binding to DNA shown here were very similar to the percentage of on-target editing that we saw. Now, at this point, particularly because of the off-target issue, we decided to do a complete wholesale change in our paradigm. And also, we wanted to test for any rescue of behaviors in the mice. So, going 3' to 5', we swapped out the hyperactive editase domain. Because I forgot to mention that almost all of the off-targets are due to rogue activity of the editase. Almost all of the non-targets had no homology with the guide whatsoever. And in fact, as we were publishing this paper, Feng and his group came to the same conclusion in a very, very comprehensive set of studies that they published in Science. So that seems, and several other investigators have now found the same. So we decided to replace the hyperactive domain with the domain from a native enzyme. Otherwise, it's the same. We swapped out the synapsin promoter for a more ubiquitous promoter, a synthetic CAG promoter. And the guide RNAs are still under control of U6. This time our control is a box B GFP construct that has just 18 nucleotides of GFP, and I'll show you later why we chose this particular control. We also changed the mutation that we used. This, instead of using R106Q, which we used before, it was in the DNA binding domain, we chose another MECP2 molecule, another mutation, W104X. It's also in the DNA binding domain, and it also loses stability and doesn't bind to DNA. We chose this mutant because we're now using the native catalytic domain, and the context of the mutant adenosine shown here, UAG, is the ideal context for editing by the endogenous enzyme. The endogenous enzyme really cares about the 5' and 3' surrounding nucleotides, and some make it less editable, and this is the ideal one. And now we even switched the serotype because Benjamin was busy and came up with a new serotype that he claimed was more widespread and better in C57 Black 6, which is what we use. So almost everything is different in this experiment. And we're not injecting directly into a brain part. We're going for systemic intravenous injection because we want to look at phenotypes. So Sanger sequencing, MECP2 function. Now we're going to look at Rett-like phenotypes and do next generation sequencing and hope that we had less off-targets. So we first looked to make sure there was RNA to be edited in this mutant, and there is. It's significantly different from wild type, but there's still plenty of RNA that can be accessible for editing. We then did a Western blot. This is whole brain now, to look at MECP2 protein levels, and in the wild type there's a band of the correct size and there's really nothing in the mutant. So this is a very unstable mutant. And so we then looked at on-target editing, which is shown here, and we dissected different regions of the brain. And I'm showing just a portion of the guide here that has the off-target, has many A's that could potentially be off-target targets. And so, because we were interested in looking at bystander off-target editing. And in yellow is the on-target A that has been mismatched with a C. And there was one real standout region which was edited, and that was brainstem. Midbrain wasn't far behind. And the other regions didn't show much editing, and I'll come back to that in a few minutes. I just want to point out that there was no bystander editing of the adenosines in the guide regions. So we then looked, I'm not showing you the immunolabeling because I've already showed it to you twice, but we then did the same confocal analysis to look at binding to DNA. We have 3 groups here. Black is wild type. Each one is a different mouse. We have 3 mice. And blue is the repaired condition. That is, there's MECP2 recovery in the nucleus and binding to heterochromatin. And green are cells in the non-repaired condition that had no MECP2 in nucleus. And their intensity measurements matched what we see in the straight non-injected mutant. And you again can see there's a very broad distribution of MECP2 intensities in the heterochromatin and the wild type. There's 150, I should say, there's 150 cells per mice in each mouse. And then in the MECP2 targeted cells, you see there's a lower level, the median scores are shown here, and again, nothing in the cells that don't have editase. And then we did statistics to show that each of these groups was statistically different from each other. So I mentioned that brainstem really had the highest level of editing, and we're kind of fortunate, serendipitous in this, because brainstem is the seat of respiratory function in humans and in mice. And so we did whole mouse plasmography in the mice to look at their respiratory rhythms. So first I should point out that we did a survival curve for these mice. Gray is a mutant, a male mutant mouse, and green is represents the mice that were injected with the GFP fragment virus, the non-targeting virus, and then blue is are the mice injected with the targeting virus. We have an N value of 16, and you can see that there is premature lethality, as we expected in the mice in these two populations, and there's an extension of living, a prolongation of living in the targeting mice, but they do eventually die prematurely. So 50% or so of the controls are dead by 11 weeks, and 50% of the targeting virus-treated mice is 17 weeks. So that kind of determined when we should do the respiratory measurements, and we did them around 8 weeks, 8 to 10 weeks, so before they started their precipitous decline. And as I said, we did whole mouse plasmography, and I'm just showing some representative samples of their breathing here. So here's wild type, very irregular breathing, no apneas. Here's a mutant, very irregular breathing and apneas. That's also true of mice injected with the control virus. And you see that the mice injected with the targeting virus look a lot better. And I know this is a very critical audience, and I know you're looking and saying, oh, wait a minute, oh, the frequency here is really not quite the same. And I think that's likely true. We haven't really looked at the frequency that much, but they look much more like the wild type clearly than these. And then the quantification for the number of apneas is shown in this panel. There's a lot of variability using this assay. This is, again, mutant mice, control mice with the control virus. This is wild-type mice, and you can see they look very much, in terms of the number of apneas, like the rescued mice. And then we also did an irregularity score, which looks very similar in pattern to the apneas. So why did we see an effect on the brainstem in particular, given that the editase, which is on the same virus, was expressed throughout the brain as predicted by the Ph.D. virus? And it made us think that maybe it was the guide that was dictating where the repair was happening. So to test that, we used a base scope analysis where we can look at the guide by in situ hybridization, and then in a dual way look at by immunohistochemistry to see where the editase is. And so the results of that analysis are shown here. First, looking at the percentage of cells that are editase positive, what really stands out is that hindbrain, or brainstem, wherever it is, brainstem here, is not very high compared to all of the other brain regions, and yet that's the region where we saw the best repair. On the other hand, if you look at where the guide is expressed, you see that it is expressed very highly in the brainstem, so that's already sort of getting a little suspicious. And then you look at the percentage of cells that are editase positive and guide positive, and now brainstem is high. In fact, this pattern looks very much like the pattern of the guide. And then when we plot a ratio of editase-positive cells to GFA-positive to guide-positive cells, you see that the ratio, which was high there, is very low here, indicating a higher percentage, indicating that it's really guide that's driving this specificity to the brainstem, which was totally unexpected because the U6 promoter is thought to be ubiquitous, but nobody had ever really tested it in the brain. Now, I have to say, we can't formally exclude that there's an element of stability involved in this, but the simplest interpretation is that there's some cell specificity attached with the U6 promoter. We then looked at off-targets, and we saw an order of magnitude less using the native domain, but still 300 sites. And here I'm showing a plot of editing with the MECP2 targeting virus versus editing with the GFP virus. You see it's a very good correlation, again pointing to the fact that the off-targets, even though they're lower, are still coming from the rogue enzyme. We therefore decided to see how toxic these off-targets might be, so we injected wild-type mice with our MECP2 targeting virus. The virus, the editase expression is throughout the brain, as we predicted, but we really couldn't tell those injected mice from wild-type mice. So their mean body weights remain the same. This is over a period of about 20 months, and they never showed any Rett phenotypes, and they were no different than the controls. Now, we didn't do a really careful, detailed phenotyping of the mice. I'm sure if we did, we might pull out some phenotypes, but it had been published that the off-targets would lead to lethality, but we didn't see any evidence for toxicity in our hands. Okay, so I just have a couple more, three more data slides. So now we're thinking of doing a wholesale change again. So we have to get rid of the off-targets, and there are a couple of ways to think about doing that. One thing is to generate more mutations in the catalytic domain that would give higher efficiency and greater specificity. And in fact, in Feng's comprehensive paper in supplementary figure 10, panel B, he actually did a mutagenesis of the catalytic domain on the hyperactive background, and he found 2 mutants which showed higher efficiency and very good specificity. And actually, I spoke with him this afternoon, and we're going to try it and see how it works on the native protein. And then another thing you can think about is forgetting about expressing a catalytic domain and just try to express a guide, much simpler, streamlined, and see if you can recruit the endogenous enzyme. And this has been batted around for, I don't know, several years at least, and nobody except Chenjie in Guoping's lab really decided to see what the ADAR distribution was in mice. So he's kindly letting me show you his unpublished data here. And so this is immunohistochemical staining with antibodies to ADAR1, ADAR2, ADAR3. And Chenjie actually had to do quite a bit of research to find a good antibody, because this is all, of course, dependent on how good the antibodies are. And so what comes out of this analysis is that ADAR1 and ADAR2 are kind of similar in being distributed throughout the brain, except for a couple of regions. One is the thalamus, which is highly enriched for ADAR2, but basically absent in terms of ADAR1. And the other is that ADAR1 is expressed better in the cortical areas than ADAR2. And ADAR3 is not expressed as well as either of them, but interestingly is expressed in the thalamus. So this, I think, gives some hope that it might be possible to recruit the endogenous enzyme. So we started a collaboration with a chemist, Thorsten Staffers, who's been working on editing for a long time, who had been thinking about using a guide-only strategy. And during the course, I have to say, during the course of all our experiments, the field is moving much more quickly than us. And so sometime in the last few years, the idea appeared to use a circularized guide instead of a linear guide, which we had been using. And Thorsten's lab, this experiment was guided by Philip Reuchling, which I know I pronounced wrong. I practiced it with him on the phone, but I gave up. But he said that Germans can't pronounce it either, even though he's German, so I didn't feel too badly about it. Anyway, he's really the head person, the head postdoc on this project. So they generated a circularized guide, which contains a region antisense to MECP2 with an AC mismatch. And then instead of a contiguous sequence of MECP2 guide sequence, they prefer having clusters of smaller sequences from 18 to 30 base pairs with bulges in between. And then the key thing for recruiting endogenous enzyme, they believe, is to include a stem loop from one of the AMPA receptors, which is highly edited by endogenous ADAR at an RG site. And then the guide also includes the ligation stem from the circularization, and it's all under control of the U6 promoter. So they first tested the circular guide versus a linear guide in transfections using a human cell line where ADAR1 is expressed. This is an isoform of ADAR1, which is constitutive in the nucleus, and in their hands, they got much better expression. So we put their guide into our system. We used phb.eb again, injected retroorbitally, waited 4 weeks, and so far, this is unpublished, we've only done Sanger sequencing. We have 3 mice, and we were pleased to see that we could see editing about the same level as in our system. Now, you'll notice that I said there's 3 mice, but sometimes there are 4 dots, and that's because sometimes we used both hemispheres. And interestingly, look where the high editing is. It's in brainstem, midbrain, and thalamus, which is the same regions that I showed you before. So I think, once again, this is a strike for the U6 promoter, because we're using the 6 promoter in both systems. Anyway, this is the first, there's only been one other experiment using the guide to attract ADAR in liver, and actually our data in the brain was better than that. So I think this is a good starting point, and we haven't looked at off-targets yet. We haven't done any behavioral analysis, but I think this is at least a good starting point to continue to think about this approach. So limitations and open questions. Does targeted editing efficiency last throughout life in mice? We don't know yet because we only treated the males, but now we can treat the females, which live a normal lifetime, and see if editing lasts throughout their lifetime. And how early should treatment begin? I'm of the opinion, and I've talked to other people today, that the treatment should, earlier the better. Well, that's something we can investigate in the mice. If we were going to stick to our system, is there an immune response to the bacteriophage protein that should be tested in non-human primates? Can editing efficiency be improved with next generation guides, particularly for hard to edit codons? I'm not going to, because of the time, I'm not going to show you the data, but we just published a paper with Pete Beal. He's an editing chemist at UC Irvine. And we were able to much more efficiently edit 270, the MECP2 mutation R270X, which in our hands, without improved guides, gives us absolutely 0 editing. So I think that's looking up. Can efficacy and specificity be improved with next generation ADAR mutants? I already talked about this a little bit, or enzymes from other species, as I've done for CRISPR. And finally, delivery is a problem. It's just a common problem in the field. Can the field generate new viral serotypes with increased potency and specificity for neurological diseases that have a broad circuitry impairment? So there's a lot of emphasis on finding serotypes that will allow infection of specific regions of the brain, but there are many diseases like Rett where the whole brain is affected, so we need a better delivery for those. I talked to Feng Cheng a bit about this, and Ben, are you Benjamin or Ben? Ben Deverman, and it sounds like this field is really expanding very, very quickly. I showed pictures, but I forgot to mention some of the names of the people who were involved. John Cinnamon really took a gamble and led this project as a postdoc. He now heads a group at Pfizer. I think they're working on genomic editing. Jack and Jenna are two technicians. Jenna did the confocal microscopy. Jack's an excellent biochemical technician. And then two postdocs, Sabrina Cragness and John McCormick, have just joined the group. We did our original AAV constructs with Hiroyuki Nakai, who's an AAV biologist. Shannon McQueenie helped us with the RNA-seq. The mutants were made at the University of Connecticut by CRISPR. I talked about Peter. I talked about Thorsten. Talked on and on about Chenjie. And I have to acknowledge Chi Chan, who did all the base scope for us, all the analysis in the OHSU pathology core, and he was just stolen away by the Brigham, which is unfortunate. And then we now make our viruses at the Penn Viral Core. This happened because of the pandemic, and Nakai's lab was shut down for quite a bit, but they seem to make very good virus. And all of the work, as I said, except for an RSRT trust to John Cinnamon, was funded by the National Institute of Health. Thanks for your attention. Happy to entertain any questions. Yeah.
I have a question. So, do you know the update on using the endogenous AMP receptor sequence to RNA editing instead of bacteriophage binding? But endogenous editing, endogenously RNA editing happens in the AMP receptor subunit.
Yes.
So I saw people were trying to use that sequence to.
Well, that's what was in the Stafford's. That was sort of the idea that was in that Stafford circularized guide. Mm-hmm. It had the RG site that's edited by endogenous ADAR.
I see. So
so it had the MECP2 sequence, but then it had a stem loop.
But that will bring the
endogenous ADAR.
Data to the sequence.
Yeah, that was the idea.
I see.
So that's the editing we get is presumably just from that, from the endogenous ADAR. We don't know if it's one or two, because actually there's some overlap in the editing.
There's no way to get rid of the bacteriophage sequence then?
We could get rid of it altogether if we can use just the guide.
Yeah, it seems like the recruiting ADAR is the endogenous ADAR is really key for those strategies, right?
Yeah.
Improving the efficiency
So
of that. That's Shape Therapeutics has been doing a lot of.
Yeah, but you know what the problem is? There's still some basic science that needs to be done because ADAR1 mainly does allele repetitive sequences. ADAR2 has a few dozen, mainly ion channels and receptors, but they can overlap. And we don't know yet what exactly is everything involved in recruiting the enzyme to their targets. We know it has to be double-stranded. We know some of them are aided by RNA binding proteins, but where the specificity is so then we don't know how to make it better with the guides. That's the problem. I mean, we can make some rational guesses based on structure and so forth, but.
U6 finding was surprising, I
Yeah.
think, because we've been able to get reasonable gene editing efficiencies, not RNA gene editing efficiencies with U6 guided guides.
But you only
all
need to
over
cut
the-
once.
What's that?
You only need to cut once.
That's true. Yeah.
That, that I think makes genome editing a little more favorable for that, than it only has to cut once. I think that's the difference.
But it also seemed like looking at the level of editing at the sequence level, maybe underreporting what the real improvements are in terms of both the behavioral rescue as well as if you just look at MECP2 protein levels and expression and
Right.
heterogeneity.
Well, and
So it may be that you're certain areas of the brain you have very high levels of total RNA editing, but even in other areas where it's much lower, the real difference is what fraction of cells are transduced versus what fraction of RNA in each cell is transduced. Those
Well,
are kind of two
that's
different
exactly
things.
right. And I think, like I said, I think we got lucky with respiration. Because we got really good coverage of the enzyme and the guide, and it happens to be the seat of respiration. So that's the thing, a lot of motor behaviors involve lots of different regions of the brain. Maybe we just don't know what the other regions are for breathing, but there certainly is a center there in brainstem.
So is that the only phenotype that was really well rescued by this approach, the motor phenotype?
And I think that's partly why the mice still died too, because we didn't rescue enough cells.
There's a question on the on Zoom. Well, it's not on Zoom, it's on YouTube. So Jonathan Alexander asks, as females are mosaic, what is the effect of these treatments in wild type cells?
Right. So that's why we injected wild type mice with our targeting virus. And we didn't see, I should have mentioned this, we didn't see any evidence for off-target editing within the guide region, and the mice seem fine. It's not as good as doing it in the female model, but in a way it's the same thing because we're injecting the wild-type cells.
Compared to the exogenous supplied ADA, is that do you think, have you done comparison, or do you think that the lower efficiency with the endogenous ADA is because of recruiting, or is it because of the amount of ADA activity?
With the guide only?
Yeah, with the guide.
So with the guide only, it was about the same as we got with the excess ADAR. And what I think is that we just need, we've only been 2 experiments done, one in liver and this one. So I think we need to spend more time on guide design.
So the amount of availability of ADA may not be an issue for them.
Well,
Is it sufficient?
I think according to Chenjie's methodology, it's not. It should be possible, except what I was saying, that we don't really understand how ADAR chooses its targets. I think that's the problem. We understand on a, kind of high level, but we don't really, there could be things in vivo that are affecting. Most experiments in RNA editing have been done in culture, not in vivo, and certainly not in the brain. So it's, I think, just showing what the limitations are so far. Brain is challenging, that's why I like it. Yeah.
Yeah, question. Sorry if I missed this, I came in a bit late. So for a cell not to have any MECP2 because of the deletion or the mutation, that's deleterious. And we also know that having a duplication or overexpression is bad. But I'm wondering, what's your view on how much is the minimum level? Because with RNA editing on a per cell basis, you're not going to get to the level that the cell would normally be at. But is like 10% editing in a cell, is that enough, or do you need 20? Is there any insight on that?
A very good question. So we don't know, just generally, how much MECP2 you need to add in different cell types to recover function. But were you here for the Adrian Bird's experiment? Yeah. So that stop sequence is actually leaky. Okay, it's not a perfect stop. And when we looked at those mice, we got those mice to repeat the experiments in our own hands. And when we looked at those mice, they actually lived quite a bit longer. And when we measured how much MECP2 leak there was, it was about 10% of MECP2. So, and other people from different contexts have sort of found the same thing, that even a modest amount of MECP2 makes the mice a lot better. So, we're not talking about a cure tomorrow or anything, but I do think that if we can get the level up significantly, either by genome editing or by gene replacement, or by RNA editing, the affected girls would be a lot better.
I'm curious if, oh, sorry.
No, I don't know, but just based on some of the mouse experiments.
So do you think that 10% read-through was just an artifact of that construct design, or because there's patients who have stop codons in their genes too, and they
but
have
I think
disease, so.
it's a problem with the design, and it's not uncommon. Yeah.
Sorry, related to that, you showed this diagram with the mutations that are very deleterious and then more sort of the polymorphisms in the general population.
Yeah.
Are any of those known to be sort of hypomorphic alleles that express like a bunch less MECP2? But
Not that I know, but there are hypomorphic mice, male mice, and they look totally fine. So they're missing 50% in each cell, and you can't tell them apart. In fact, they were originally scored by Adrian as wild type. And then he discovered actually that they were hypomorphs. So somewhere between 10 and 50%. I'm not saying you get a perfect cure or anything, but you greatly improve, I think, the day-to-day life of the girls.
Can we go for reception?
Thank you.